Alright, taking a look at part 2, we're going to talk about the special case of water and the movement or osmosis that's going to be taking place. Now, if I take a look here, you can see at this aquaporin, this is the specialized protein that allows water to pass through from an area of high water to an area of low water, which we are going to call osmosis. So that's that process going to be taking place. Now, osmosis is the diffusion of water. Really, it's movement of high to low, but of water. So diffusion and osmosis are different. Diffusion really is movement of a solute from high to low. Osmosis is a movement of water from high to low. But as long as you remember diffusion is referring to high to low, and wherever there's more pure water, it's going to go where there's less pure water to equalize that amount of equilibrium. That's what's going to take place. Now, 3 terms that I'm going to have you guys make sure you understand the differences between hyper, hypo, and iso. Hyper is more, hypo less, and iso equal. Now, in that same sense, they'll utilize those as our structures and components. If I have 3 different solutions, okay, each has a cell in it. This first one has a lot of solutes. When I'm going to say these X's refer to sugar. The second one has a lot of the X's on the inside. The third one has the same amount on either side. Now, when I look at this material, I'm going to refer to it as what are my solution and what's my cell considered. Solution and cell considered. Okay, now the solution, since it has more materials within it, the solution has more materials in it than the other, the solution is considered hypertonic. Since the cell has less, it is hypotonic. So usually in our prefixes. This one, the cell, has more, so it's hyper. The solution has less, so that's hypo. These are both equal, so that's going to be the same. The same for those. Now, what can be said is that if something, in our case, if we go back to our solution, we had a cell, I've got a lot of stuff out here. This may be hypertonic because I have more materials. This is hypotonic, meaning I have less sugar, but in the same sense that hypotonic is got more water, so it's hyperosmotic. Whereas the hypertonic is got less pure water, so that's hypoosmotic. So you can imagine that the one that is equal, the isotonic, is also isoosmotic. So looking at our structure, if I put in material, water into a normal freshwater structure versus balanced versus salt water, what's going to happen is that the normal water is going to rush in faster, causing the cell to burst on this one. If it's balanced, it's going to stay equal. But if I put more water, had more water in the cell, the water is going to leave the cell, causing the cell to shrink. So, kind of an example of which is going to take place is in plant cell. So if I have maybe a piece of lettuce leaf in salt water, I've got a lot of salt out here. This lettuce, the water is going to rush out faster, so this cell is going to shrink because the water is going to leave faster than the stuff can come in. Opposite of that is if I have something like a raisin, and it's got more sugar in it, the water is going to rush in faster, making this cell expand. Okay, hyper, hypo, and iso, so that's what there's kind of get into. Turgor pressure. Turgor pressure, I didn't show you that, but this is the pressure that's going to be showing that allows the cell membrane. So if you look at this structure here, the cell membrane is allowed it to push itself to the edges, so it's really pressing all the way to the edges by the water vacuole inside, the stored vacuole, pushing it off the side, so keeping the pressure. That's its normal turgor. Now, if I release water, that pressure starts to decrease, and as that pressure starts to decrease, the plant will start to wilt, and so we don't want that. We want to keep the increased turgor pressure. Now, if I need to get rid of water, I can help regulate that. Some organelles can do that. Some structures do that, like an example of paramecium, which is a protist. It has a contractile vacuole, which can actually expel water. So here's an example of where that turgor pressure is decreased because the water left. It shrunk that central vacuole, so the cell membrane no longer could keep that constant pressure on the cell wall, so the whole inside shrunk, and that made the cell kind of shrivel up. And that cell shrinking or wilting in a plant is known as plasmolysis, the cell membrane coming off of the cell wall. Water being able to pass through quicker, water is much smaller, but remember water is polar, so it cannot actually go through the lipid tails that are going to be found within our cell membrane structure. So if I do that cell membrane here, it can't go through here. It actually has to go through a protein, but it can go through much faster because they are very small than most of the other things. Now when you look at the cell membrane, it is more than just a simple barrier. It's more than just those Dippin' Dots. It is a mosaic. There's a whole bunch of stuff scattered around because they have lots of different functions, from transport to signaling, transduction to attachment points to, I mean, you name it. There's a lot of different things going on onto it. 1972, Singer and Nicholson had developed the model of the fluid mosaic model, versus Davison Bernoulli had developed one that looked more, let's see if I even have it on here. It doesn't look like it's even in here. But Davison Bernoulli model had one where it just talked about the proteins only on the surface on either side. But in that one, it didn't account for how I can get materials through. So we knew it can't be that way. So Singer's model is actually the correct model of the fluid mosaic model. Okay, it's a whole bunch of different materials within it, because what they're going to do, these proteins can serve different functions. You will want to know these three different types of proteins. There's peripheral, that is just on the outsides. That can be one on the top or on the bottom. So here's an example of a peripheral one there. Okay, an integral protein is actually one that's just embedded. Okay, it's one that's embedded in, doesn't have to go all the way through. And a transmembrane protein is an integral protein that is all the way through. So it's one that goes all the way from the top outside the cell to inside the cell. They play a key role in cell-to-cell recognition, allowing cells to interact, signal to each other, important organelle tissue development, so cells working together make tissues, tissues to organs, so on and so forth. Now, example or ABO blood grouping, we have proteins on the surface of the blood cells that actually signal. So if you have A blood or B blood, or in the codominant multiple allele trait, AB blood. Other jobs that these functions or proteins in the cell membrane can do: they can formulate, they can allow for transport, they can allow for being enzymes. So something will come in, substrate will come in, and two things will come out. It can be a signal, so something can attach onto it and then send the information down. This may be an example of the nervous system, where I have a neurotransmitter hitting this information and it sends a signal on to the next neuron. Intercellular joining, where I communicate between two different proteins, maybe even pass materials through those. Cell-to-cell recognition, so I have an actual cell land on it. This would be a great example of a white blood cell attaching or adhering to helping with destroying of other cells, like any foreign debris. An attachment to cytoskeleton, other cellular matrix. Okay. Osmosis really just comes down to, again, the movement of water from high to low. Okay. The rest of this is all done. So that is the last portion of part 2 on the cell membrane.